Okay, so we're going to start our afternoon session, and our first speaker is Karel Svoboda.
Thank you very much for this generous, expansive introduction. I've only known him for about 15 years, so it's, there's what is there to say? Anyway, so this is a great conference. I'm really happy to be here. The science is spectacular, and it's really an honor to be able to tell you a little bit about our work. So I'll focus on multi-regional circuits in the mammalian brain. As you know, even the simplest behaviors involve multiple brain regions. For example, the pupillary reflex, a very simple behavior. Light enters the retina, is processed by the retina. Signals related to light intensity are then passed on to pretectal nuclei, then on to the Edinger-Westphal nucleus, also in the tectome, then to the postganglion, to the ciliary ganglion at the back of your eye, right? Motor-related signals, and then nerve fibers coming out of the ganglion then innervate the ciliary sphincter to control pupil size. So in multiple stages, sensory signals are converted to a movement. And so you might wonder, why does even such a simple behavior require multi-regional neural circuit? And the answer, of course, is likely that pupil size is modulated in multiple behavioral contexts in a flexible way. Now, in general, learned, flexible, and cognitive behaviors also involve multiple brain regions. In fact, we think that, and we're still counting, that the mammalian brain contains about 1,000 brain regions. Each brain region, in turn, contains multiple cell types. And I think cell type-specific analysis is something that even this group eventually will have to deal with. This is not something I will touch on today. So classically, though, of course, neurophysiological experiments has focused on a few neurons recorded in a single brain region. And I think for these reasons, we have relatively little understanding. You're supposed to change the, I don't know if this is, yeah. This is the right slide. This is a black slide. So anyways, we have relatively little poor understanding of how brain regions interact and how the entire brain produces behavior. In our lab, together with multiple collaborating laboratories, we're trying to explore how multiple brain regions combine to support prototype cognitive behaviors. So I'll tell you a few stories in the context of a simple decision-making task. So 8 years ago, we developed a delayed response task for head-restrained mice. This is a decision-making task involving a short-term memory component. So during the instruction epoch, also sometimes referred to as sample epoch, mice perform a sensory discrimination, either an auditory or tactile discrimination, and mice indicate the decision with skilled directional licking about the sound quality or the tactile stimulus. But mice are trained to respond after delay epoch lasting seconds. Okay? So during the delay epoch, the mouse has to rely on internally generated and maintained brain activity to bridge sensory instruction in the past and its action in the future. It's a kind of short-term perspective, short-term memory. And because the mouse remembers what to do next, we refer to this short-term memory as movement preparation. It also relates to preparing movements. It's very similar to delayed memory-gated saccade tasks, for example, that folks have studied very elegantly in non-human primates. Now, behavioral experiments show that mice use the delay epoch to plan fast and accurate directional licking. So, for example, if we shorten the delay epoch, the subsequent movements become slower and less accurate. Now, today I mostly focus on neural activity during the delay epoch, so that the activity that links the instruction epoch and the peri-movement epoch, the behavior reporting. And a few years ago, we identified the brain regions in the frontal cortex that is critical for maintaining this short-term memory or motor preparation. And we refer to this as the anterior lateral motor cortex, and I think Mike Shadlin will have more to say about the anterior lateral motor cortex later in the context of a working memory task, a different kind of short-term memory. So we've recorded from this region and encountered neurons with slow and selective dynamics during the delay epoch. This is here an example neuron. On top are spike rasters. We color code right movement trials blue, left movement trials red. And on the bottom are the mean spike rates. And this neuron exhibits activity that is maintained after the instruction is gone. And this persistent activity is selective for the upcoming movement, okay? And for this reason, we refer to this memory-related activity as preparatory activity. It relates to the future movement. So it turns out that most neurons in ALM exhibit this kind of preparatory activity. Intermingled neurons show selectivity for rightward movements, so that's blue above red here, or for left movements, red above blue, and these are intermingled, these neurons. And importantly, during the delay epoch, activity of these neurons is monotonic and typically ramps up or ramps down up to the onset of a movement. And these neurons, as far as we can tell, are intermingled in the ALM. And in this cartoon, what you see neurons that are right selective and left selective, right selective blue, left selective red, intermingled in the ALM. Moreover, similar selectivity is seen on both sides of the ALM, okay, during movement preparation. And both the sides of the ALM are connected by powerful projections, the corpus callosum, which is very strongly expressed in these frontal motor cortical areas. In a series of studies, we've shown that the detailed dynamics of preparatory activity in ALM during the delay epoch is critically important for the mouse to remember what movement to do next and to prepare upcoming movements. And I'll outline the kinds of analysis we've done and just touch on a couple of different experiments. So we'd like to relate activity of neural populations in ALM to behavior and record with silicon probes, typically 30 neurons simultaneously. So there's a 30-dimensional activity space. So the neural activity defines a 30-dimensional space. And the activity of each neuron corresponds to the direction in this space. So population activity then traces out a trajectory, on average, a different trajectory for right movement trials and left movement trials. Now we define a choice dimension, a choice direction in this activity space as the direction that optimally separates activity anticipating the different movement directions. So that's the choice direction, and then we can, this is a simple dimensionality reduction method, we can project the activity along this choice direction. And in the direction, in this choice direction, trajectories separate in this ramping fashion during the delay epoch. So the choice dimension, it turns out, contains most of the direction selective activity in the system. And there are a couple other modes. So this is low dimensional activity. So Eric Shea Brown argued earlier that this might be because we're looking at a trained network and at the output end of a categorization network, perhaps explaining this low dimensional activity in ALM here during decision making. So, and it turns out that activity in the choice dimension explains directional tongue movement. So, so in the next few slides, I'll show neural population activity along this choice dimension. So and how do we know that these neural trajectories here have meaning? So by construction, of course, the average trajectory in the choice direction correlates with behavior on average. This is how we've constructed these trajectories. But the mouse, of course, has to use single trial information for behavior. And single trial trajectories differ from the average trajectory. And it turns out that the detailed structure of the trajectories on individual trials predicts the behavior, including reaction time and the probability of moving to particular directions, which I'll show you here. So here, what I've shown you is that the activity during the delay epoch, here in this cartoon, and the detailed location of the activity trajectory at the end of the delay epoch predicts the direction of movements. So for movement trials, where the trajectory on individual trials is close to the average right trajectory, mice make right movements with high probability on right movement trials. For trajectories that sneak into, that leak into the territory of the left trajectories, mice are more likely to make left movements subsequently, and the converse is true for left movement trials. So activity during the delay epoch predicts movement direction on a trial-by-trial manner. Now, does this by itself imply that ALM activity is participates is critical for planning movement? Well, we're looking here at a correlation. There's, of course, the formal possibility that among the many brain regions that we don't observe at this time, there's a critical brain regions that triggers behavior that also feeds fluctuations into ALM, and we're looking at these fluctuations in ALM. So to link behavior and neural activity in ALM more closely, we manipulate activity in ALM and observe what happens both to the activity trajectories in ALM along the choice direction and behavior. And I'll just show you one experiment of several. It's a crude experiment, but I think quite powerful experiment because I think it shows that ALM activity is critically linked to planning of future movements. So here what we're doing is we're inactivating both sides of ALM transiently at the beginning of the delay epoch, seconds before movement onset. So activity is essentially silenced, and afterwards, after the inactivation, activity recovers, but the trajectories lose all memory about trial types. So they meander here along the choice direction and activity space. Moreover, the behavioral reporting is now random. Animals equally likely move to the left or to the right after the end of the delay epoch. But on a single trial level, we can still predict the movement direction based on the trajectory location during the delay epoch along the choice direction. So we think that this experiment is interpretable because aspects of the activity trajectories recover in a non-random manner, presumably because of the connectivity in the network. And so by analyzing the effects of various kinds of perturbations on both neurodynamics and behavior, we've convinced ourselves that preparatory activity in ALM directs future movements. So I've shown you that ALM shows persistent and selective preparatory activity and that populations of ALM neurons direct future movements. So in the next 20 minutes or so, I'll show you how ALM actually interacts with multiple brain regions during the delay epoch to produce preparatory activity and decision making. So let's consider these multi-regional interactions involving ALM. So why look at multi-regional interactions? So this picture is simply to convince you that ALM is part of a multi-regional circuit. Of course, you didn't need convincing. These are a bunch of single neuron reconstructions in ALM. The somata of these neurons are roughly around here where the arrow is. And you can see different neurons are plotted in different colors. You can see neurons that project across the corpus callosum to contralateral ALM, strong callosal projections, not just to the cortex, but also to the underlying striatum, both in the contralateral hemisphere and the ipsilateral side. Other projections from ALM go to thalamic nuclei, in particular the VM thalamic nucleus and MD. Furthermore, neurons from ALM project to midbrain nuclei, in particular the midbrain reticular nucleus and the superior colliculus, then also to the basal pontine nucleus, which connects ALM to cerebellar circuits, and then strong projections to contralateral parts of the medulla that control orofacial behaviors, like the movement of the tongue. And in fact, other people have described ALM as essentially an orofacial motor cortex. So based on these strong projections to different brain regions, it's not surprising that preparatory activity is not restricted to ALM. We, in fact, found qualitatively similar preparatory activity intermingled left and right cell activity in multiple connected regions in the brain, including the thalamus here in the middle of the brain, the basal ganglia, in particular the substantia nigra reticulata, and the superior colliculus. And similar observation has been made actually in other species, in non-human primates, and in the context of different kinds of short-term memory behaviors, including working memory tasks. Now, at the same time, this activity is not everywhere. It's sparsely distributed in the brain. For example, ALM shows preparatory activity, but neighboring regions in the motor cortex do not, okay? So it's a sparse subset of brain areas that express preparatory activity, and this is suggesting that a distributed network that is selectively coupled produces preparatory activity. And it stands to reason that perhaps short-term memory in general is likely an emergent property of a neural circuit distributed across multiple brain regions. So the kinds of questions we'd like to know: what are the core substrates of decision-making in short-term memory? What are is the distributed, redundant distribution of preparatory activity perhaps a mechanism for robustness, where one brain region helps another brain region recover from perturbations? What are the causal relationships among brain regions showing similar dynamics? And is the coupling between brain regions static, or does it change during different phases of behavior? Is there gating in communications between brain regions? And the kind of approach that we're taking is to examine this distributed network by multi-electrode recording in one or couple of these brain regions, and then using optogenetics to silence or activate in a calibrated manner one brain region while measuring neural activity in other brain regions. Okay? In other words, we perturb specific network nodes while monitoring the activity, the network response to the perturbation. Sort of a simple kind of engineering experiment. So such perturbation experiments have already revealed a rich repertoire of multi-regional interactions. I'll give you 3 different examples, 2 very quickly because they're published, and then I'll dwell on the third one for a little bit longer. So I'll start with the ALM interhemispheric interaction, this connection across the corpus callosum. Preparatory activities expressed on both sides of ALM. And as mentioned, if we silence activity in one hemisphere, this is just to show that we silence both abolish selectivity and silence activity in one hemisphere in this single neuron, PSDH. Different neurons do exactly the same thing. It's silenced by photoinhibition. Activity and selectivity in the other hemisphere is essentially unchanged. So despite this strong anatomic coupling, the 2 hemispheres are organized into seemingly independent modules. And each module maintains activity and selectivity independently. Okay? Interestingly, after the end of the perturbation, so when we turn off the perturbation, activity and selectivity recovers. See that this neuron recovers its selectivity to what it would have been in the absence of a perturbation. And it turns out that information from the intact module flows into the perturbed module to recover the information in the perturbed module. And this self-corrective feedback makes memory robust to perturbations. Now, other connected brain regions connected to ALM show completely different kind of coupling. And here, let's focus on the thalamus. ALM projects to small parts of the thalamus, parts of VM and MD, and those same parts of the thalamus project back to ALM in a reciprocal manner. In fact, the same neurons that project to ALM receive input back from the thalamus. So, and it turns out if we silence activity in the thalamus, activity is completely abolished in ALM. So what you see here is activity with photoinhibition in the thalamus compared to control activity. You can see that all these points corresponding to units hug the x-axis, meaning 0 spikes. So in the absence of input from the thalamus, this frontal cortical area cannot maintain activity or selectivity, of course. And the converse is also true. Okay, so if we silence ALM and record from the thalamic regions that receive input from ALM, we see that activity is greatly diminished by about 70%. In fact, we think there's an underestimate because we only silence a part of the region that provides input to these thalamic regions. So in contrast to the interhemispheric interaction, thalamocortical connections are critical for driving persistent activity in this thalamocortical loop. There's tight coupling between thalamus and ALM, and therefore we think of ALM and the connected thalamus essentially as one module. Okay, the thalamus essentially acts as an extension of cortex, and so whenever I say ALM does this or that, I really mean ALM, the connected parts of thalamus do this or that. And as I already mentioned, the thalamus is in the middle of the brain and connects cortex to a number of subcortical regions, including the basal ganglia, the midbrain, and the cerebellum. And ALM also projects through various brain regions to the thalamus. So There are long-range loops here that we're only beginning to explore. All right, so the last example is about dynamic interactions between sensory cortex and ALM. And this is actually, I have to confess, this is the work in an abstract that we submitted to Cosyne, and the commissars, based on coin flipping, rejected it. Yesterday I was so there's a bit of a rogue talk here now. But I noticed that we're very much on the top of the soft threshold, so that's something to be proud of here, so it's good. I'm happier now, so yeah. So the flow of the signals in the brain must be the subject of multiple controls as the behavior unfolds over time. And as has been shown in many psychophysical experiments, the brain has the ability to respond to stimuli at some times and then tune out the same stimuli at other times. For example, sensory stimuli playing during the delay epoch, during the instruction epoch, have great influence on neural activity and behavior. After all, they instruct the animal what to do, but the same stimuli could have much smaller impact when presented at other times, for example, during the delay epoch. After a decision has been made, the memory should perhaps become insensitive to the stimuli that triggered the memory. So the question really is, where are the knobs controlling the flow of information from sensory areas to these premotor areas, and how do they work? Now, as you know, sensory information in the mammalian brain is presented in the sensory cortex, and we'd like to know if robustness of short-term memory during the delay epoch is in part due to gating of information flow between sensory cortex and ALM. But in a sensory discrimination experiment, the representation of the sensory stimulus could be gated in a number of regions, okay, downstream of the sensory cortex. For example, in the thalamus, there's quite a few lovely studies showing that the thalamus helps to gate salient sensory stimuli. So to isolate communication between sensory cortex and ALM, we need a constant stimulus in sensory cortex in terms of the spikes in a defined ensemble of neurons. And so Arseny Finkelstein and Mike Economo came up with I think, elegant experiment to do this, to probe interactions between sensory cortex and ALM directly. Instead of sensory stimuli, here in their task might simply detect a short train of optogenic stimuli for water reward, and the photostimuli were applied directly to the somatosensory cortex. So there's in a transgenic animal that expresses channel rhopsin in layer 4 neurons in the somatosensory cortex, shown right here, and that's the input side of the somatosensory cortex. And so, therefore, the spikes under the experimenter's control and the same stimuli can be administered any time during the behavior to probe coupling between sensory cortex and ALM. So let's now, during this task, where animals detect direct stimulation of the somatosensory cortex during the instruction epoch, let's record neural activity first in sensory cortex and then in ALM. So in the sensory cortex, neural activity is actually qualitatively similar to behavior with actual tactile stimuli. Here are, this shows 4 example neurons recorded in the somatosensory cortex. These neurons show brief phasic spiking responses to photostimulation, no hint of short-term memory or preparatory activity. Stimulus-evoked activity rapidly decays to baseline conditions, and none of the somatosensory neurons show memory-related activity that spans the delay up to the movement onset. And like with tactile information, we see lots of neurons that are modulated up with photostimulation and others that are modulated down, but all show brief phasic responses. And this is very much in contrast to ALM. Now we're recording on the same condition in ALM downstream. Here, a subset of neurons also show phasic activity to the photostimulus, but it's a small subset, small fraction, less than 10%. And this activity is inherited from sensory cortex. The majority of neurons show memory-related activity that bridges the photostimulus and the onset of movement, similar to the persistent preparatory activity seen in the standard sensory discrimination task. So here, see a neuron that ramps up after stimulus detection, up to movement onset. Another neuron here that ramps down, that is left movement selective, this is right movement selective, and so on and so forth. Very similar to the condition with the sensory discrimination task. Now, the same stimulus can now be applied at different times during the behavior, for example, during the delay epoch. So let's first look at the behavior. As instructed, mice detected and behaviorally responded to the photostimulus when applied during the instruction epoch. After the mice were rewarded to do so, right? The same stimuli now applied during the delay epoch had a progressively smaller effect. Still caused some changes early during the delay epoch, but caused no changes late in the delay epoch. So therefore, transmission of sensory signals to circuits mediating memory and decision-making, motor planning is filtered in some sense, or gated with time. Should mention a similar behavioral experiment was performed by Seidema and Newsome in the context of the famous random dots paradigm in macaques. They used microstimulation in MT to bias behavior. All of you are probably familiar with this famous behavior. They applied microstimuli before, during, and after the visual stimulus. During the visual stimulation, microstimulation showed the classic effect, biasing of the behavior. When microstimulation was applied after the photostimulus, the effect was much smaller. Okay, similar to our experiment and consistent with some kind of gating between MT and downstream areas. So we can now ask, what are the mechanisms of gating in the system? So one hypothesis, and this was my favorite, is that the photostimuli cause diminishing activity in ALM over time, perhaps via presynaptic inhibition, or because of mechanism in the somatosensory cortex. But synapses, presynaptic terminals in the brain, have a plethora of G-protein coupled receptors that are activated by various neurotransmitters that can modulate synaptic transmission greatly. So there's a biophysical substrate for gating at the sensory cortex to ALM synapse. So in this hypothesis, in ALM, we'd expect large stimulus-rated signals during the instruction epoch and progressively smaller signals in the delay epoch, mirroring the behavioral effects, right? And so, but the data largely contradict this hypothesis. So here, down here, the graph shows stimulus-related population activity now recorded in ALM. This is during the instruction epoch. This is projection of the population activity along a direction that is most modulated by the stimulus, here during the instruction epoch, and then different parts of the delay, and the modulation is essentially unchanged. The little attenuation that you've seen here actually mirrors a similar attenuation of optogenetic activity in the somatosensory cortex. This implies that sensory information is not gated on its way to ALM. Instead, we find that gating happens at the level of ALM dynamics. And over time, preparatory activity in ALM becomes robust to the incoming stimulus. So let me discuss this, make some predictions in the context of a model which essentially provides a picture of what's going on. So this model was developed together with Sandro Romani, PI at Genilia, and Lorenzo Fontola, and a postdoc in Sandro's lab. And Lorenzo is actually here, and he'd be happy to discuss details of this work, as would I. So this model, it's an attractor model, really captures behavior and neurodynamics in the delayed response decision-making tasks that I've introduced earlier. And I should have mentioned also the very important contributions of Hidehiko Inagaki, and then previous versions of the model by Shaul Bruckmann and Kevor Day. So this model captures many features of the data, and the constraints are based on electrophysiology, many types of optogenetic perturbations directly applied to ALM in that case, and behavior. So in this model, ALM neurons that share similar kind of preferences connect to each other and compete through GABAergic neurons. And this is just one module on one side of the ALM. There are actually two modules coupled through the corpus callosum that are not shown here for simplicity. And the neuron, this circuit receives two types of inputs, very importantly. One is the selective sensory input, which tells the circuit really what to do, tells the animal what to do, and a non-selective ramping input that is related to the learned timing in the task. Okay, this ramping input is shallower for longer delay epochs and steeper for shorter delay epochs. This ramping input in this model produces multiple attractors. On a baseline condition, there's a single fixed point in ALM activity. The ramping input ramps up two attractors, or depending on the number of movement direction in this behavior, two attractors or two fixed points show up. And as the ramping input increases, it pushes these attractors apart, further apart over time. And it's this moving the attractors apart over time which produces robust preparatory activity. And the sensory stimulus, what it does, it biases the system into one or the other attractor early in the dynamics, in instruction epoch, and then once the system is trapped in one or the other fixed points, these fixed points are pushed apart by the non-selective ramping activity. So by moving the fixed points, the ramping input really explains the slow time constant related to the task structure, which is on the order of seconds. That's why we need the ramping input. We have many types of evidence that there's a ramping input coming to the ALM. In contrast, this is to the time constant of the intrinsic dynamics of the thalamocortical circuit, which is much faster, on the order of hundreds of milliseconds. So if we apply perturbations to the circuit directly, we find that recovery is on the order of hundreds of milliseconds, reflecting a decay of the network to a fixed point. So these two time constants, one time constant is controlled by the ramping input. So this model could also explain gating. So let's focus on choice-related activity here in a cartoon. These activity trajectories correspond to the fixed points of the attractor model: right movements in blue, left movements in red, or anticipating right movements in blue, anticipating left movements in red. The photostimulus, detection of the photostimulus in the instruction epoch, moves the attractor, causes big changes the system from the default attractor into the blue attractor, okay? Associated with rightward movements, the retrodirectory corresponds to no stimulus in left movements. And as the attractors and trajectories separate, the deflections now produced by photostimuli subsequently often fail to move the trajectory over the separatrix, indicated here as a dashed line. So after the perturbations, at least on correct trials, the trajectory relaxes into the default fixed point, and this would correspond to correct trials. But early during the delay epoch, the photostimulus still causes changes in behavior on occasion, this is then expected to also be associated with large changes in the trajectory. So these are essentially the predictions here, and this is essentially the behavior that we see in the data. So here is the activity along the choice mode for right movement trials and correct trials, and left movement trials here in the detection task. And you can see that in correct trials, photostimulation during the delay epoch caused only transient perturbation of the trajectory that perturbed to the red trajectory, decay to the correct fixed points. On error trials, the trajectory switch to now resemble the right movement trials, so essentially flips in a bistable manner to the other attractors. So that shows, I think, that attractor dynamics can gate information flow across brain regions. So this working model that I've shown you is sort of algorithmic. It encapsulates the key principles by which the system operates. And the principles are decision-making via discrete attractor dynamics, and a pitchfork bifurcation that is driven by ramping input. But compared to real neural networks, of course, there are huge simplifications at work. Connectivity is deterministic. The circuit is highly structured. All neurons of one type essentially share the same rates and dynamics. And the circuit is, of course, specialized to perform a particular task. Now, Lorenzo has been exploring data-driven models implemented using training recurrent networks. I know you've heard a lot about this over the last few years in these meetings. So these models, in contrast to the conceptual models I've shown you, can show rich dynamics, diverse spike rates, and it's been shown that these networks can, of course, simultaneously solve multiple tasks. So in our incarnation here, the network receives 3 types of inputs. One transient input corresponding to the photostimulus applied to the somatosensory cortex. Context related to the task, auditory stimuli that delineate the instruction epoch, and non-selective ramping input. And we find that this ramping input is critical to make the models work, consistent with the data. So the recurrent network model is trained to produce now the observed neural dynamics. Here we rely on methods that were pioneered by Kanaka Rajan in a paper 2-3 years ago. And here each neuron has a private target function. Each unit in the network has a private target function that corresponds to peristimulus time histograms for different trial types. And then synapses are adjusted in this network using force learning to match the target function. And training is done in the presence of noise. So this figure illustrates that the recurrent network learns the target function. On top are the target functions for 3 neurons, 3 exemplar peristimulus time histograms, a delay selective neuron, here neurons that show transient activity during the photostimulus, and down here the responses of neurons after training of the recurrent network. And now we can analyze the dynamics of this network to stimuli that it has never seen. Sorry, skipped one. So here, the choice-related activity looks similar for the, essentially identical for the recurrent network in the data, and this is by construction, so the blue curve on top and the red curve on the bottom. But like the mouse, the network has never seen perturbations applied to the somatosensory cortex during the delay epoch. But again, on correct trials, the system decays to the retrodirectory with fast dynamics. And on a subset of trials, it produces error trials by switching to the other trajectory. So the network has arrived at a solution very similar to the conceptual attractor model with these discrete attractors, 2 discrete attractors essentially moving apart in time. We can now look at the role of the ramping in this, and this is my last slide. I think I'm also running out of time. My last data slide, at the ramping in the behavior and in the model. Okay, so we'll first look at the behavior similar to the behavioral experiments. The recurrent network model shows limited robustness early during the delay epoch and a lot more robustness late in the delay epoch. That is because these attractors move apart in time. And then in individual trials, we can ask what the slope of the ramping is by analyzing groups of neurons, looking at the steepness of the ramping. We can do the same in the network model. In the network model, we vary the ramping from trial to trial. We do this during the early delay epoch, and we find that when the ramping is steeper, when the ramping is more rapid, the system is more robust, and the escape from the default attractor is less likely than when the ramping is shallower or weaker. So this shows that the ramping here separates attractors and is a determinant of robustness. So what I've shown you is that diverse types of functional coupling between brain regions, these brain regions really interact in fundamentally different ways, and the mechanisms thereof, I think, are very interesting topics for future investigation. Coupling across brain regions can, of course, enable connectivity, robustness, novel computations, and coupling can be gated to route information in flexible ways, and attractor dynamics can gate information flow across brain regions. Just want to mention that we're only scratching the surface of a rich biology of multi-regional interactions. There's clearly a lot to learn, and we think that in the mouse brain, we can now probe dynamics perhaps in a comprehensive manner. And so we're collaborating with multiple other laboratories in a project funded by the Simons Foundation to do this. So this project relies on standardized experiments in short-term memory behaviors and analysis of many brain regions simultaneously using multiple neuropixel probe recordings simultaneously. And we're trying to do this both using prospective short-term memory tasks like the one I've shown you and retrospective working memory tasks like the one you'll hear from about later. And if you're interested in this kind of endeavor, let me know. So let me finish up by, again, highlighting the folks who've been key players in this endeavor. So No Li and Zhenkai Guo, who are now, who run their own labs at Baylor and Tsinghua, started work on motor preparation in the lab and developed this task. Hidehiko Inagaki and did a lot on attractor dynamics, and he recently did a paper that you might want to take a look at if you're interested in this. Sandro Romani and Lorenzo Fontellani did the modeling, and Arseny Finkelstein and Mike Economo, who for some reason dropped off, developed the optogenetic detection task and did the routing experiment. And I thank you for your attention and take questions if there are any, and if you have time.
Questions?
Hello? Yeah, great. Thanks a lot for the talk. I was wondering about the experiments where you have those trajectories that are getting far apart from each other during the delay time. How would it look like when the mouse is, he wasn't trained on the specific delay time that you are showing? And specifically, if you give him longer time delay than the one there he was trained on? Thank you.
Yeah, so there's a literature on this in non-human primates, and we've done experiments also in this task, both varying the delay epoch and making the delay epoch unpredictable. So if you vary the delay epoch and the animal knows what to expect, the ramping essentially follows the duration of the length of the delay epoch. Shallower ramping for longer delay epoch, steeper ramping for shorter delay epochs. And if you make the ramping unpredictable, in general we see very fast ramping and then a steady state. Yeah.
How long can this steady state go on for?
This is sort of we've sort of pushed it to 10 seconds or so. Mice don't like to do that. So for that reason, we typically operate with 1 to 2 seconds. Yeah, good. Be happy to discuss these
One
issues.
question only, each, please.
Yeah, so I was wondering, one thing that shows up in your data but doesn't seem to show up in the model is that the activity in the choice direction seems to cross the separatrix during the stimulation during the early delay period. But in the model, it seems like, the external source is just sculpting the energy landscape, but it just depends on where ALM is. And I was wondering if you think that like switchover that seems to still reliably occur during the perturbation during the delay epoch is like implemented by something else, or if you think that you're just missing something by using the choice direction.
Yeah. I think this is really this is a very, very good eyes, and it really depends on what kind of weighting you use in the projection. And I don't think that's fundamental, but it's really the plot that I showed you was designed to highlight the perturbation. So if I showed you strictly the choice dimension, you wouldn't see any of the stimulus-dependent perturbation would be much smaller.
I see.
But there's sort of an excitation inhibition transient that you see in that projection. Yeah.
Last question. Yeah.
Thank you. Yeah, I was fascinated by the non-selective ramping activity. So most of the ramping activity in the brain is in fact kind of selective and anticipating outcomes or actions.
Yeah.
and actually, scales with things related to reward statistics and uncertainty. So I'm wondering exactly what kinds of the nature of the kind of ramping activity that your model requires, and how that squares with some of the ramping activity that's been observed thus far.
So the ramping activity in ALM is largely selective, but there are places in the brain where you see mostly non-selective ramping activity. So for example, in the output nuclei of the cerebellum, in the vestigial nucleus connects to ALM through the thalamus and shows ramping activity that shows very little selectivity. If you transiently silence the vestigial nucleus, you actually abolish selectivity in ALM. So
selectivity to the action.
Selectivity for the action, yeah. But the key experiment, there are probably other players. Neuromodulation also ramps in a non-selective manner, we think, and there's data from other laboratories, including dopamine. So there could be many mechanisms that sculpt this urgency signal that then drives this selective activity in the cortico-thalamocortical loop.
Okay, let's thank Karel.